Okay, HL, let's do our SL notes for B2.2. I'm going to do these in two parts. I'm going to be referring to the slides that are available. I'll be doing kind of like a base level of notes and referring you back to slides for some of them, just to reference the things you don't remember. Let's start off with the very top here, cell theory. Cell theory, this can be found on slides 1 through 7 of the SL 2.2 notes. So if you want some more details to annotate to this or need some deeper explanations, you can find a lot of information there. All right, so the cell theory, a theory is something that is rooted in a lot of evidence, but in science we never say things are ultimate truths because we keep discovering new things and would hate to change a truth. So theory is what we as high as we go as far as certainty. There's three parts to the cell theory that serve as a sort of a big understanding for how life on Earth is. The first one is that all living things are composed of cells. Composed of cells. The second is that the cell is the basic unit of life. And third is that cells come from pre-existing cells. This is a little different than the characteristics that describe a living thing, which we'll get to later on. This is more about how cells are related to living things, not what life is. So we assume that you're already living. Let's go into more detail now about just like what does that mean with respect to cells. We've got there's some more details on slides 1 through 7 if you want to add to any of that. But if you think that gives you a pretty fine understanding, you remember that from last year, then you are good to go. All cells on planet Earth can be classified as either prokaryotic or eukaryotic. Prokaryotes, there's two classifications of prokaryotes. You are either a bacteria, and a bacteria is something that has a cell wall with something called peptidoglycan in it. Or you're an archaea. Archaea do not have a cell wall with peptidoglycan in it, and quite often archaeas are termed as extremophiles because they live in very intense environments like high salt environments or very acidic environments or very hot environments and things like that. There's a lot of interesting classifications of extreme environments. We don't need to go into any more detail than that, though. And then eukaryotic cells include cells that are found in, this one's pretty easy, plants, but you can guess the next one, animals, fungi, and there's a fourth one, things called protists. If they don't fall into one of those three categories, kind of lump them into protists there. You may be familiar with things like amoeba or paramecium. Those things are protists. Okay. There's some differences between prokaryotes and eukaryotes that you need to know. We'll go into that a little bit later. Let's talk now about the structure of a prokaryotic cell. Now, you do have to know specifically that we are talking about what's called a gram-positive prokaryotic cell. A gram-positive prokaryotic cell has a thick layer of cell wall. Let's see, where should we label that? We don't need to label that anywhere just yet. It has a thick cell wall all along the outside, followed by a membrane underneath it. It's called gram-positive because gram refers to a particular kind of stain that you can add to the outside of it, and all bacteria are either gram-positive or gram-negative. And when scientists are trying to identify a bacteria, the first thing they do is a gram stain to see what kind it is, to sort of narrow it down to just one category. Gram-negative bacteria have a much thinner cell wall, and they have a second membrane on the outside of the cell wall, which is just a little bit different. All right, let's get right into it. Let's start off with the capsule. The capsule is a super sticky layer of polysaccharides that helps it adhere to surfaces and sometimes keeps it from dehydrating as well. I starred that to mean that not all bacteria have a capsule. Okay. Next up is right underneath. This is something that all bacteria have, and that is a cell wall. And in bacteria, this cell wall is made of that substance that I talked about just up top called peptidoglycan. And then just underneath the cell wall is the plasma membrane. All prokaryotes have a plasma membrane. And just like our plasma membrane in our eukaryotic cell, it controls what comes in and out of the cell. All right. All prokaryotes have, let's switch colors here, cytoplasm, which is that watery liquid that everything floats in. And there's a lot of metabolic reactions that happen inside of that cytoplasm. Metabolic reactions happen inside that cytoplasm. You know, think about when we learned about the electron transport chain, and remember that the mitochondria is full of a fluid on the inside, and that's where the Krebs cycle happens, kind of similar to that. Okay. Speaking of reactions inside of our prokaryote, there are ribosomes, and you know what ribosomes stand for. Yeah, I mean, not stand for, but you know what they do. They synthesize or build proteins. Now, proteins. Ribosomes in a prokaryote are what are called 70S ribosomes. It means that they're a little smaller than eukaryotic ones. Eukaryotic ones are 80S ribosomes. The S stands for Svedberg units, which is the guy who sort of figured out that there was a difference between them. His method of measurement resulted in, you know, one going to 70 and one going to 80. I don't know how he decided what those units would be, but that's what it is. Next up, I'm going to skip all the way down here to the bottom. There we go, all the way down here to the bottom. This is what we call our nucleoid region. And this is where the DNA is. This DNA is circular. We talked about that when we talked about replication, which means it's just one big, giant, jumbly loop. It is also naked, which means there are, gasp, no histones. So it is all completely unwound. But I mean, it's significantly smaller than our eukaryotic DNA, so it doesn't need to be as organized as ours. I skipped down to that DNA because right over here, we've got a little tiny piece of DNA called a plasmid. This is a little circular piece of DNA and generally has the information in it for one single trait. One single trait. These little pieces of DNA can be exchanged between bacteria, so they can pick up new traits. Okay. Two more things. These two things are a little similar to each other. They both stick out of the cell, and they are not found on all prokaryotes. First, these little guys here are pili. They look like little whiskers or little hairs. They help the cell attach to surfaces. Sometimes they're also used to swap DNA with other things, with other prokaryotes. And then finally, we have our, also not found in every single prokaryote, we have our flagella. I should have said flagellum since this is singular. Flagella is plural. And this is movement, helps the prokaryote get around. So all of this information can be found in slides 9 through 13. There's more detail on each of those about these different parts. This is like a basic level of understanding. And then finally, you have to be able to identify 3 of these parts on a micrograph. This is what's called a micrograph. This is a fancy way to say an image from a microscope. It's one of those kind of European ways that they say it. All right, it's pretty easy to see the nucleoid region here. This is the nucleoid region. This is the naked circular DNA. It's easy right here. You can see this cell wall. It's kind of thick. And if you were asked to label the cell membrane, you would need to point at this inner layer of the cell wall. So this inner layer right here, this is the plasma membrane. Controls what comes in and out of the cell. Okay, now I want to skip ahead a little bit and talk about something that is common to all life. We're just going to focus on prokaryotes in this particular video, but there are things that are common to all life, including prokaryotes, so we'll address that in this as well. We're going to start with slide 57 here. And over here, let's see, let me get my pointer on. So I'm going to be reading this sentence right here, and that is that all living things, even unicellular organisms, are able to do the following processes of life. So remember I said earlier that we would get to what all things in life can do. That's this list right here. Now, I'm not going to go over all 8 of these things because I think a few of these things are pretty easy to get. Movement, I think, is pretty easy to get. All living things do some kind of motion or able to do some kind of transfer of themselves from one place to another. All living things grow, reproduce. All living things have to give off their waste products somehow. All living things have to get some kind of nutrition inside of them. They do it in a lot of different ways. The 2 that I do want to just mention really fast are first, homeostasis. Homeostasis is a way to say that it keeps its internal environments in a certain specific range. Keep internal environments in a range. So think about your own bodies. Your own bodies maintain a specific body temperature, a specific level of blood glucose, lots of different levels of other molecules. That is all considered to be homeostasis. And when that range is disrupted, problems can happen on the inside of these cells. Finally, the other thing I want to mention is response to stimuli. And that is when the environment changes, all living things do some kind of response to it in order to, it's like a survival mechanism. So if your environment gets cold, your response to that change is to maybe stand the hairs up on your arm, to shiver, to dilate your blood vessels to increase blood flow, which helps to increase heat. Okay? And then last thing, all living things carry out these particular 8 features. Also, on our next line down, all living things contain these certain structures. No matter whether you're talking about a prokaryote or a eukaryote, whether it's an archaea or a bacteria or a fungus or a red panda or an amoeba, they always have these specific things. First of all, they all use DNA as their genetic material to pass on information to their offspring. Always there's a cytoplasm, which is that liquid mainly composed of water, where there's a lot of chemical reactions and things move around. And all cells are surrounded by a plasma membrane made of lipids. And remember that lipids have the hydrophilic head, the hydrophobic tails. And when something has both of those things, it is called amphipathic. I know you were taught amphiphilic last year. They do mean the same thing. IB will call it amphipathic. But if you can remember that amphi kind of means the same thing, then you're good to go. Okay, that's it for part 1 of the SL notes. Tune in for part 2, where we will go through eukaryotes, and there are some other SL standards related to microscopes. We will do those in class. Okay, that's it.